We're going to take a look at endo and exocytosis in this lecture. In vesicular transport, we're going to be using a vesicle, a membrane-bound sac, to move things into and out of the cells. So we're going to use this to move larger molecules like proteins, and this is an active process, so we do need energy to do endo and exocytosis. Remember, endocytosis is when we're moving things into the cells, and there's a couple different types. And exocytosis is when we're moving things out of the cell. We'll start with endocytosis. And one of the types of endocytosis is called phagocytosis. This is done by some of the white blood cells in our body. Phago means to eat, and cyte means cell. So it literally means cell eating. So the white blood cell is going to find some sort of solid particle, maybe it's a bacterial cell or it could be dirt in your wound or something like that, and it's going to surround it with its membrane and then take that inside the cell and pinch its membrane off. Okay, so now you've got this vesicle, this little membrane-bound sac surrounding whatever it's taken inside. And then the lysosome, one of our organelles, will fuse with the phagosome, and the digestive enzymes in the lysosome will then break down and digest whatever was ingested by the white blood cell. Phagocytosis is not specific, so the white blood cell is going to eat anything foreign. It doesn't care if it's E. coli bacteria or salmonella bacteria. It's going to eat whatever it finds, so it's not very specific. And this is just showing you an animation showing the membrane surrounding the solid particle, maybe a bacterial cell, and then we've got the lysosome fusing and releasing the digestive enzymes and then digesting whatever was taken in. Pinocytosis is cell drinking. Pino means to drink, and all of our body cells will do this. So they're going to just take in some of the extracellular fluid and whatever's dissolved out there, so whatever solutes, and they'll just take that in and they're just sampling their environment to see what's out there. And so all of our cells will do this. And again, it's non-specific. So just whatever is in the extracellular fluid, that's what gets taken in. And the last type of endocytosis is very different from phagocytosis and pinocytosis. This is called receptor-mediated endocytosis. So this one's different because it's very specific in what gets taken into the cell. So we have some specific receptors that are out on the cell membrane. And for example, we take in the LDL, the bad cholesterol, into our body cells by receptor-mediated endocytosis. So these receptors will just bind to the LDL, the bad cholesterol, and when it binds, that will stimulate some proteins on the inside of the membrane to start folding, and that will bring the membrane in and pinch off. And that's how you bring in the LDL, those low-density lipoproteins, into your body cells. So this one's very different because it's very specific what is brought into the cells, whereas phagocytosis and pinocytosis are not specific. Exocytosis is the opposite of endocytosis. So we're going to be moving these larger molecules to the outside of the cell. So for example, maybe this is a pancreas cell, and we've got the protein hormone insulin, which is stored in these secretory vesicles, these little membrane-bound sacs. And then when the pancreas gets the signal to release the insulin, the vesicle will fuse with the cell membrane, become part of that, and then the insulin will be dumped out into the extracellular fluid. So we're going to see exocytosis also being used to release neurotransmitters in neurons, and we also release antibodies from white blood cells this way too. The last thing to talk about is transporting molecules across the epithelial barrier. And just some vocabulary words. When we take a look at the epithelial barrier, we've got the apical membrane, which is going to face into the lumen. And then the basolateral membrane of the epithelial cell is going to face the blood. And then in between the epithelial cells, we have tight junctions, which remember those are leak-proof barriers. So that's going to prevent things from moving from the lumen in between these epithelial cells and into the extracellular fluid. Because it would be really bad if this was, for example, the intestine, and we had the bacteria in the GI tract moving through into our abdominal cavity. That would actually kill us. Let's take a look at the small intestine and how we absorb glucose from our diet and get it into the bloodstream. So there's actually a lot of stuff that goes on. If we look on the bottom on the basolateral membrane of this intestinal epithelial cell, we have a sodium-potassium pump. And remember, that's primary active transport. And we know it's primary active transport because the sodium is moving from low to high concentration. It's going up its gradient. And potassium is moving from low to high concentration, also going up its gradient. And we're directly using ATP in our picture here. So we know that's got to be primary active transport. Right to the left of that, we have some potassium diffusing through a potassium leak channel. And this is passive transport, right? So diffusion through channels, we're going to move from high to low concentration. So we're going to go down the gradient. So we don't need any energy to do that. And then moving glucose across this basolateral membrane. This is facilitated diffusion. So we're moving from high to low concentration. And we're using a carrier protein. You can't see it, but it would change shape as it flips back and forth. So that would be facilitated diffusion to move the glucose across. On the apical membrane, we have secondary active transport. So we know that because the sodium is moving from high to low concentration, so it's going down its gradient, and glucose is moving from low to high concentration, so glucose is going up the gradient. So whenever you have one molecule going down the gradient and the other one moving up the gradient, then you know that's got to be secondary active transport. So besides moving the solutes across the epithelial barrier in the intestine, we also need to absorb water from our diet as well. So kind of a good rule of thumb to remember is that water is always going to follow the solutes. So here we have some solute molecules, so maybe for example glucose or other things, and we're moving those from the lumen of the intestine into the extracellular fluid here, this interstitial fluid, and eventually that'll get moved into the blood. And because we're moving solutes into the interstitial fluid, water is going to follow. So water always follows the solutes. So wherever the solutes are going, water is going to follow by osmosis. An example of a disease that's caused by membrane transport not working correctly is cystic fibrosis. So this is an inherited recessive genetic disorder. And what happens is they're not able to move the chloride ions into the mucus and the airways like they should. So if we're moving our chloride ions out into the mucus, these sodium ions follow whatever the chloride ions are doing. And water, remember, follows the solutes. So because you're moving the sodium and chloride ions out into the mucus, water follows by osmosis, and that will make the mucus more watery and easier for the cilia to move that mucus with the trapped bacteria and things that you might have breathed in up to the back of your throat, where you either will cough it out or swallow it. But in cystic fibrosis, the chloride pump does not get put out there, and so the chloride ions are not moved out into the mucus. And if chloride ions are not moving, sodium ions aren't going to go anywhere. And if you're not moving any solutes out into the mucus, water's not going to follow the solutes. So the mucus ends up being too thick, and the cilia are not able to move that mucus with the trapped bacteria and things you've breathed in. And so that just sits in the lungs. And then people with cystic fibrosis are very prone to getting pneumonia. There's a nice animation here that just shows what happens when the mucus gets thick and how the cilia are not able to move. So if you want to watch that at home, it's a pretty good animation. And the last thing to look at is called transcytosis. So transcytosis is going to be used to move larger molecules like proteins across an epithelial barrier. So for example, maybe we have, and I know this is not exactly correct picture, but maybe we've got our epithelial layer here that's making up the lining of our blood vessel, and we need to move the insulin hormone into the blood. So we're going to go from the extracellular fluid outside this pancreatic cell and then move this across the epithelial barrier and into the blood. So in order to get these large molecules across these epithelial barriers, they're too big to fit through, pumps or carrier proteins or channels. So we're going to be using endocytosis on one side, and then on the opposite side we'll use exocytosis. So it's called transcytosis because trans just means across.